Why aren't we finding things that are bigger and better? Like if you look at the mouse longevity literature, the longest, non-developmental intervention for lifespan is a caloric restriction experiment done 40 years ago.
Just because we age doesn't mean we have to age. Think about the plasticity of lifespan across divergent species that had a common ancestor. You can see these enormous effects, right? Say that you want to compare the genomes of diverged species with very different lifespans. I think we only just now are starting to really have the tools to do that compellingly.
I'm going to read off the name of the intervention, and I want you to rank it from 1 to 5. My favorite, rapamycin. My name is Matt Kaeberlein, and welcome to the OptiSpan YouTube channel. Hey everyone, welcome to the OptiSpan podcast. Today's episode is the first in a series of interviews that I conducted while attending the 2024 American Aging Association conference in Madison, Wisconsin. I'm thrilled to present this discussion with Dr. Mark McCormick from the University of New Mexico in Albuquerque. Mark did his PhD thesis research with Cynthia Kenyon, one of the true luminaries in the field, and then went and did his postdoctoral research studying the genetics of cellular aging with Dr. Brian Kennedy. Mark and I have published several peer-reviewed papers together, including what really represents the culmination of more than a decade's work carrying out an unbiased genome-wide screen of the genetics of longevity for budding yeast. Mark is a rigorous, talented scientist and a fantastic mentor to his trainees. So in today's episode, we're going to do a deep dive into some of the cutting edge research that is going on right now in Mark's laboratory, where they have been exploring a new class of longevity drugs that act by inhibiting tRNA synthetase enzymes. This work has already shown promising results across a variety of different model organisms, including budding yeast, nematode worms, and mammalian cells. We discuss some of the intricate mechanisms for how these inhibitors work. We spend a lot of time talking about the challenges of translating this research into practical applications and the exciting potential for future discoveries. So sit back, relax, and join us for a captivating conversation with Dr. Mark McCormick as we explore the frontiers of aging research and what it could mean for extending health span and lifespan in humans. All right. Hey, everyone. Coming to you live from the 2024 American Aging Association meeting. We're going to try something a little bit different. So this is completely unscripted. I'm sitting down with Mark McCormick, and we're going to see where this conversation goes. So Mark is an assistant professor at the University of New Mexico in Albuquerque, did his PhD thesis with Cynthia Kenyon, and then postdoc with Brian Kennedy. So he comes from a strong line of classic aging researchers, so to speak. So it's great to get to catch up here at the meeting, and we'll just see where this conversation goes. So why don't you tell us a little bit about what you've been doing in your lab in terms of what are you most excited about in your research?
So during my postdoc in Brian Kennedy's lab, in collaboration with your lab, so you kind of know this work, one of the things that came out of it is that this transcription factor in yeast, GCN4, played an important role in lifespan, and that was the transcription factor itself, functionally, in its whole pathway, is kind of conserved through humans.
Right.
So one of the things we started to do was look at other ways to upregulate this transcription factor to see if we could recapitulate this effect on lifespan. And some of the things we've done is look at tRNA synthetase inhibitors. So initially what we found is that mutations that affect the ribosome could lead to upregulation of this transcription factor. And really just going back to the literature, people showed that if you have problems with these tRNA synthetases, usually they showed it genetically with mutants that aren't too good, you could also upregulate this thing. And then we found in the literature that there are these great, highly specific inhibitors chemically of these enzymes.
Right.
So we threw them on there. And to summarize a little bit of work, we were able to show an increase in replicative lifespan in yeast using some of these compounds, and an increase in lifespan in Caenorhabditis elegans using some of these compounds. And the most recent stuff that we've published about them is we went into mouse embryonic fibroblasts, and we showed that these drugs can sort of do all of the same things to all of the same parts of this pathway. And so we didn't really think about lifespan of these cells in a dish.
Sure.
But one of the newer things that we found, sort of maybe not shockingly, is that these drugs that probably convince cells that they're amino acid starved, because tRNA synthetases sort of attach amino acids to tRNAs.
Right. Maybe it's worth just taking a minute to go through what are tRNA synthetases and what do they do, and how does that tie into the protein synthesis translation pathway?
Yeah, there's a whole.
Yeah, I know it's complicated.
Well, there's a potential controversy there. I would say there's more than one model, and they're not necessarily mutually exclusive, But so when cells want to make proteins, what they do is every little piece of DNA that tells it an amino acid that it's supposed to sort of Lego together to make a protein, there's a tRNA unique to that amino acid that brings that guy in to stack them up and make proteins.
Right.
And then it goes back to grab another amino acid and sort of cycles around.
And this is at the level of actually making the protein. Again, for people who aren't in the biology world, DNA to RNA to protein. So the RNA goes to the ribosome. That's where the action happens to connect these building blocks up. And you have to have these tRNA synthetases that will actually bring the constituent amino acids to the ribosome to build the protein.
That's right. And so you have like at least 20 of these guys, because every amino acid has a tRNA and they each have their own tRNA synthetase. There's actually a little more to it, because your mitochondria kind of have some of their own crazy ones that are entirely separate, which comes into our story in at a level we maybe won't dig into. But so you have a lot of these enzymes, and it turns out that tRNA synthetases are a very ancient and highly conserved part of how everything makes proteins.
Right.
And as a result, soil microorganisms that are constantly trying to murder themselves, each other with chemical warfare, that have been a great source of a lot of antibiotics, produce a lot of highly specific toxins that inhibit a single tRNA synthetase in, some neighbor of theirs that they've got beef with.
Right.
And so we just went and dug out a bunch of those.
Which is sort of just as an interesting aside. I mean, there's lots of important molecules that fall into that pattern, where these are made by certain microbes as a way of biochemical warfare to outcompete their competitors, rapamycin being another example, right? Produced by different mechanism, completely different mechanism. But
and to not to rain on anyone's parade, but what is often the case is that in the lab you can try to chemically modify these compounds, and you almost always just make them worse, because they've been under evolutionary selection for
or less effective, maybe is the way to
years.
say it. Yeah.
Yes. And so it's hard to do a better job than the ones you can find often. But we have at this point quite a few. So I think in our most recent paper we're up to maybe 7.
Okay.
And they all appear, the thing they have in common is that they inhibit this one enzyme, right? And for most of them, it appears to be that there's not much chance they do anything else. Maybe not for all of them.
Sure.
And so, if you're a cell and these tRNAs are not being connected to their amino acids.
So that's what the tRNA synthetase does, just to be clear, because I kind of was confusing in the way I explained it, right?
Yeah, so you got to reattach an amino acid to these guys, and then they keep showing up with these amino acids, kind of conveying them in one after the other, or you can't make proteins, which is, can't make anything.
Right. So do all of these different drugs inhibit the same enzyme?
No. In fact, none of them I don't think any of the ones we have inhibit the same enzyme. In some other stuff we're doing now, we're starting to hit some of the
different flavors of tRNA synthetase.
I think maybe in the paper 2 of them are the same. But long story short, it definitely works with more than one of these guys.
Okay. And so just to take a step back again, the reason why you started looking at these was because you knew GCN4 was important in yeast, and that the homologs of GCN4 in other organisms seem to be important for longevity.
It wasn't actually known
at that point.
I don't think the longevity connection was made outside of yeast then.
Okay.
But so we went straight into, as soon as these drugs extended in yeast, we were like, well, let's dump them on some other guys.
Yeah. Okay. So that's super interesting, actually. But just again, to give context. So the reason, and I know you know this, but for the people watching, the reason why GCN4 we thought was interesting came from work that Brian and I did when I was a postdoc, where we found that a whole bunch of mutations in ribosomal proteins, so components of the ribosome itself, increased lifespan in yeast. We didn't know what the mechanism was, but we, through a series of studies, converged on GCN4 being one factor that was activated by these ribosomal defects, and found that that was important for the lifespan extension. And then you kind of took over that research, finished the genome-wide screen, published that paper, and started looking at these drugs that targeted the protein synthesis pathway through a different mechanism, and found that they also increased lifespan.
Right, because I'm sure that some of these drugs had been shown. The things we saw right away were people had shown that mutants in the tRNA synthetases that kind of lower their activity clearly shot up GCN4 and its orthologs, and that had been shown in multiple systems.
Got it. So you knew that those were good targets to increase GCN4.
And they knew these drugs basically recapitulated that, and I think it had been shown to some extent. Outside of the context of longevity and aging, this stuff has been pretty extensively studied and well studied. Sometimes you get into a field and it's kind of a mess, but there's a lot of nice, careful work for us to sort of stand on top of to keep going. So that made it easy to start finding some of these compounds. And I'll say of the ones we've tested, they haven't all worked in the dose ranges we've been able to get into solution and throw on things, but quite a few of them have. So the cell's thinking that it's, so to come back to where we branched off, and we can kind of go however, the cell probably thinks you need to eat more protein, man, because coming up short on amino acids.
Right.
So interestingly, we did a thing where we did some RNA-seq in the MEFs. So we looked at the levels of every single gene's expression to try to see what sort of patterns might explain the changes we're observing, right? And then we did the same sorts of things in yeast. And something that jumps out is autophagy, which is where the cell sort of gobbles up its own components to recycle them, is way up. And so is the proteasome. So imagine you're trying to build something out of Legos, but you're running out. The castle you made yesterday may have to get broken down because you need that brick for the new thing. That's kind of autophagy and the proteasome to some extent. And so that sort of jumped out at us from an unbiased way of coming at this, but it's appealing because you might think, if cells think they're hungry for amino acids, why would that be good for them in terms of making these organisms live longer?
Right.
Maybe when they're tearing things down to recycle them, what they're tearing down is something kind of old and dusty, and then they make a new shiny new one, and maybe by sort of forcing them to do that, because they're not actually starving, they can remake stuff no problem.
So does the lifespan effect require autophagy?
We haven't published something to conclude that, but I would be surprised if it doesn't. So the problem we've had in looking at it a little is that messing up autophagy is very bad for these organisms. Right. So what you really want is a way that it doesn't hurt them normally, but it prevents this from helping. And that's been hard to get a clean result on. And so it's something we're chipping away at.
Well, and I will say for my recollection and experience in yeast, it seemed like the evidence that autophagy itself was driving lifespan extension was pretty weak compared to C. elegans and Drosophila and mice, whereas the proteasome came out early on. If you could activate the proteasome, that was sufficient to increase lifespan in yeast.
I shouldn't forget to say, if I'm not remembering this wrong, Jessica Tyler showed they just overexpressed GCN4, and I think they then deleted ATG5 or something, and it doesn't. So they directly overexpressed GCN4 in yeast. They got an effect, and I think they could show that one way of kind of crippling autophagy kind of brought that back. Okay, so there is a result out there in yeast. Yeah. So I mean, that's not in these compounds, but it's suggestive. Right.
Okay. Cool. So you had these data in yeast that these compounds extended lifespan. Obviously, even people who work in yeast are a little bit skeptical that something that extends lifespan in yeast is gonna increase lifespan in a multicellular organism and eventually up into mice. So you guys, this is nice because you've got a compound, so it's kind of the easy button to put it on worms. You have a lot of experience working in worms from your PhD, so your lab set up to do those experiments. So you guys tried that and
works great.
Works great. Like when you say works great, what's the magnitude? Are we talking
for the
about?
mean, it's maybe 60%.
Okay, so that's pretty good.
And That's what we've published, and
Yeah.
we're chipping away still, I would say.
Okay.
So
And have you
I think
done,
we'll do better.
have you done a lot of the obvious genetic experiments? Like, does it require DAF16, which is an insulin pathway? Does it interact?
So we
with-
haven't done every canonical worm pathway, actually. So the things we did a few things. Trying to think which we definitely did. So obviously there is a worm that's deleted for ATF4, which is the worm guide that's the same as GCN4.
Right. And how clear is that? Is there no question ATF4 is the GCN4 ortholog?
Not in my mind.
Okay, fair enough.
I was talking to
someone. You're the guest. You can get whatever answer you want.
Yeah, so I was just learning yesterday from someone that apparently there are people who might suggest ATSF-FS1 as a possible GCN4, but
so I don't know. Yeah, I guess I should comment.
So I need to go back and look.
Intuitively, to me, that doesn't make a lot of sense, but I haven't looked into the rationale. So
yeah, so it's on my to-do list to see where I may have missed in thinking about that. But I would say that. Let me say this. I think that if you look at a pretty strong literature outside of aging, people would say GCN4 is ATF4, like in mammalian cells.
Yeah, and I think Rich Miller and others have kind of honed in on ATF4 in mice as being potentially important for some of these longevity pathways. Yeah,
to look. So
there's smoke there. I don't know if it's a open and shut case yet, but it seems like there's some evidence suggesting this is a conserved longevity mechanism.
Yeah, and there's a few things you could say. One, sort of for the specialists, this may be cut out to the general audience, is that this whole pathway, it goes from, say, GCN2 to GCN4 to EIF2 to, it goes GCN2 to EIF2 to ATF4. Right? All the sequence is quite homologous for every part of the pathway in every organism, except the transcription factor in yeast. Right. GCN4 is kind of a weird one. It's functionally clearly sits in the same spot, but it doesn't look as similar. Whereas the worm one and the mammalian one are quite similar. And
I mean, but I do think one of the compelling cases for ATF4 and GCN4 being functional homologs and orthologs, this is again for people who like to get in the weeds, which I think many of the people watching this podcast do, is there is this really sort of unique mechanism of regulation of expression of the protein that involves what are called upstream inhibitory open reading frames, right? That are nutrient sensitive. So even if the actual sequence of the protein isn't highly conserved, there's this really sort of oddball mechanism that it's regulated by. And many of the targets that GCN4 and ATF regulate are overlapping or the same across organisms. So it seems pretty plausible that.
And I would say, even in our hands, specifically in tRNA synthetase inhibitor treated, we have RNA-seq in yeast and in mammalian cells similar.
Oh, that's cool.
And if I take the drugs that induce GCN4 in a dose-dependent manner and in a GCN2-dependent manner in yeast, they induce ATF4 in a dose-dependent manner and in a GCN2-dependent manner in mammalian cells.
Right.
So I feel like we're building a case that there could be other contenders that do something similar, but I think we're looking at the right conservation.
Okay, so this is probably a good time to say don't start taking tRNA synthetase inhibitors.
No, because So I'll say here's how we find out a good dose. We just put in as much as we can, and it kills things because eventually
you need your tRNA.
Eventually, there's like one amino acid that you can no longer use, and you can't make any protein.
Yeah. Okay.
And if we come back maybe 20-fold from that or something, we're back into a range where we can see all kinds of cool stuff.
Right. So I want to talk about mouse experiments. I know you haven't done the longevity experiments yet. That's an obvious place to go. So it'd be good to talk about like how you're thinking about that. But one question that comes to mind is there's, I think, pretty good evidence that for longevity in mice, if you restrict certain amino acids, so branched chain amino acids, tryptophan, methionine, you can get lifespan extension. Do you think that these molecules are acting through specific amino acids, or do you think it's more general than that?
I think it's more general than that. I think that my model now is uncharged tRNAs are sensed by this guy GCN2. Its whole job is to find uncharged tRNAs. And if there's any preference in which uncharged tRNAs, it's because GCN2 has a subtle preference that, it hasn't been or that has perhaps
Okay.
been noted.
And again, just to
I think,
tie this together for people, it's the tRNA synthetase that takes a tRNA from uncharged to charged.
Right. So uncharged means there's no amino acid.
Right.
Charged means we've put on the amino acid, and we're blocking the enzyme that does that. So now we have all these guys sitting around waiting for amino acid they're never going to get.
Right.
And then your cells have this whole sensor, and it looks just like the synthetases. It looks for uncharged tRNAs, but it doesn't fix it. It just notes it, and it can sense all of them. I don't know if it's equally, happy about all of them in terms of what it's going to do next.
So how does this tie together then? Or maybe it doesn't. It's fine if you just want to say, I don't know. But how does this tie together with the branched chain amino acid, methionine restriction, tryptophan restriction literature?
Well, I can tell you one interesting thing that we did note is that it should be the case. Well, this is a bit technical, so maybe this gets cut for but if you inhibit methionine charging, you can't initiate protein synthesis.
Right.
And this URF mechanism should give you more GCN4.
Okay.
Without GCN2.
So methionine may be uniquely sensitive because of its role as the first amino acid in every protein.
It can skip the GCN2 step because it turns out what GCN2 does is phosphorylated AF2.
Right.
You don't initiate, you pass the URFs.
Got it. Okay.
And we found that for one of the methionines that we tested, so for everything we tested normally, if you get rid of GCN2, it's indistinguishable from you never put the drug in there at all.
Got it.
For one of the thionine ones, there's a little bump, right, where you would get the peak, but even in the GCN2 knockout. So it looks statistically significant. So we may be observing that if you inhibit methionine charging, there's a GCN2 independent way to come right back to the same place, GCN4 or ATF4. So that's perhaps of interest in terms of some of the literature out there about methionine, but I haven't thought through
it's speculative, but reasonable. And I mean, the thing with the branched chain amino acids, again, a little bit speculative, but those do seem to preferentially activate mTOR. So that may be another way to kind of go a different route to increasing autophagy, increasing turnover of damaged proteins, things like that.
Yeah, I think that's absolutely right. And so,
okay, so mouse experiments. Why haven't you guys done it yet?
So I would say that a big part of it is that if we look in the yeast and in the worms, if we had just tried a couple doses, we could easily have not seen an effect. We have the luxury there of being able to try quite a few closely spaced doses.
So are you saying that for lifespan, there's a pretty tight dose response?
Yeah, I think, the window between when it's bad news and when you get a good lifespan is quite large. But I would say that, a tenfold change in dose would get rid of the lifespan. If you don't have a huge window to get the optimum effect. And that seems to correspond with when we look at the upregulation of ATF4, let's say by a reporter, and in the MEPS we did it both by a translational reporter for the ATF4 itself, and we made a ATF4 transcription reporter, so something that should normally transcribe. And both of them kind of have a little, because what starts to happen is you close down translation overall more than you upregulate ATF4, and you kind of cancel out your effect. Something that I should mention, because I haven't really thought about what to do with it, is that we did click chemistry in the worms to see how their translation is going. And these happy, active, very long-lived worms have much lower translation overall.
That doesn't really surprise me. Yeah, I mean, especially as adults. I think where you'd see it, right, would be in reproduction, maybe development to adulthood, and then reproduction early on in adulthood.
Yeah, and so these drugs cause developmental delay. Worms are exquisitely sensitive, and so, Yeah,
and I mean, I think that's not uncommon for some of these worm mutants that hit the translation pathway, that it seems like that. But then as adults, they look beautiful, right?
And we can do it as adult only, and it works. It just
interesting.
If that's 60% of their life, it's about 60% of the effect. It works for development only, it works for adult only, but it's a bigger effect if you do the whole thing.
And have you tried later onset in adulthood in worms?
No, and what I really want to do is take them on and off. There's a bunch of things. And one thing we did do, I sort of, you asked about other worm mutants.
Yeah.
One that we wanted to be sure to look at was E2. Mm-hmm. It extends it about the same amount it extends.
So E2 is a caloric restriction model, genetic model of caloric restriction.
It's like they can't eat as much as they would like.
Right.
And
so say that again, it's about the same as E2?
The percent increase in E2.
But what about when you combine them?
That's what I mean. The percent increase of these drugs on E2 is similar to the percent increase that it would have on the normal worms.
Which you would typically interpret, at least the first interpretation would be a different pathway than caloric restriction in that case.
I think our simplest interpretation is this is not indistinguishable from caloric restriction. It's not entirely the same thing at all. But
interesting. And do you know, do these drugs require DAF16 for the lifespan extension?
I don't think we looked. Yeah,
Come on, man.
Because I was like,
Cynthia's going to watch this. And she's going to be very disappointed.
So we will go back through. So we want. There's a lot of things we could do in C. elegans that we haven't done. We kind of went straight to
I get it.
to the BEFs.
Okay, so the mouse experiments, though, if I understood correctly, what you were saying is you want to have a better understanding of the best option for dosing to give you the best chance at seeing a lifespan extension before starting on this.
But what I really want to do is start a cohort with more doses so that at the end, if we don't see an extension, it won't be because we missed the optimal dose.
Yeah, okay.
Because I think that really puts a damper on the next thing.
So what do you need to do that experiment?
A little more funding.
How much? Because there's people watching this that they potentially could fund this project if they think it's a cool project.
Yeah, I mean, I think we run pretty lean setups, so I think it's not outrageous.
Just come on, put a number out there. How much do you need? $500,000?
Yeah, I would do it.
I mean, so the idea here would and what are your thoughts on adult onset? Right. So in other words, do you have any reason to think that it's more or less likely to work if you started the treatment at 20 months of age in a mouse? Thinking that, those are faster experiments. You can buy aged mice from JAX. That would be the preferable way to do it, unless there's some reason to think that it probably wouldn't work.
Yeah. So, I mean, my sort of answer around what you asked is like, if we were considering that, I would do that in worms because we know that answer in 3 or 4 weeks. So we haven't started them at middle age.
I got to say though, the worm adult stuff, I'm still not at all convinced that if a drug does or doesn't increase lifespan starting late in adulthood in worms tells you anything about whether it's likely to have that effect in mice.
They're post-mitotic, they're post-
and they stop eating after about day 10 or 12 of adulthood, so getting delivery of the drug.
Little.
Okay, mostly stop eating, right?
I mean, a lot less.
You know what I mean? It's a technically challenging experiment, and I'm not sure that it really, just because the life history of worms is completely different than mammals, that it gives us much insight there.
I think our best. So this is, given Rich Miller's results, I think it's highly likely that some of these drugs will extend mouse lifespan at the right dose.
Yeah.
And so I think our best chance to see that on the first go would be start from basically early.
So it sounds like you need to put in an ITP proposal for this.
Potentially.
Yeah. Okay.
So there's a few ways to do. So I would say we've exhausted most of the ones that are known that don't cost an arm and a leg. We have a few we could still add to our sort of constellation there.
Yeah. Okay. I mean, I think this is a good example of something that I am seeing more and more, which is that there are really interesting interventions out there that are ready to be tested in mice, but there just isn't the capacity, the infrastructure. The ITP can only test 5 to 7 new interventions every year. I know we've got at least 20 good ones in this current cycle because I'm on the committee that picks them.
Sure.
So I think there's a need for resources and infrastructure to allow people like you to do these experiments much more rapidly. Otherwise, we'll be sitting here 2 years from now and having the same conversation.
Yeah, and I think, these are potentially translatable in a reasonably direct form, but they also offer a lot of insight into things because we have proteasome upregulators basically, which are actually kind of hard to come by.
Right.
And so they're very of great interest for a lot of reasons. These are autophagy upregulators, maybe not so hard to come by. And we can try to tease apart whether that's the crucial reason that these things can live 60% longer.
Right.
Whether or not these are metabolized in the liver in the mice and they don't give us the same effect, or lots of other things you can run into. I think there's a lot we could sort of tease apart with them in mice. Yeah. And, and even in the cells in the worms. So.
Yeah. All right. So that has been fascinating. So now I've got a long list here. You can see the list, but you can't actually see it. So these are interventions. I'm going to try to do this with everybody here and see how it goes. So I'm going to read off the name of the intervention, and I want you to rank it from like 1 to 5. 1 being, yes, definitely has a positive effect on the biology of aging, lifespan and or health span. 5 being, no, I don't believe it at all. And 3 somewhere in the middle, like maybe. Okay.
Am I allowed to?
Yeah, you can pass.
Discretion.
You can also give a brief commentary if there's something you want to say about your answer. So we're going to do this both for laboratory animals and people. Okay. So let's start with we'll do each intervention. Start with laboratory animals, then with person.
Am I the first victim of?
You are the very first one. So this is kind of see how it goes. All right. We don't have to go through the whole list because I just kept coming up with more ideas. But I'm going to start with my favorite, rapamycin, laboratory animals.
Yeah, obviously.
1. Is that what you mean?
It's
1 to 5.
It's a clean result. Which one's the good one?
1 is good. It's like NIH grants. 1 is the best. 5 is the worst.
Good. It's like golf.
Yeah. Okay. Rapamycin. 1 laboratory animals.
Lab, yeah.
People.
1 if you got the dose right, I think. Maybe 2, but I think the dose is everything with rapamycin.
Okay. Cool. Part of the reason why I'm doing this is I don't think most people out there know the opinions of actual scientists in the field because they hear nonsense from nonsense people. Right. They don't actually know what real scientists think. So
I think, like if I take a step way back and, like, does 1 mean it definitely does something or does it definitely do something huge? Like, I think
we're just targeting the biology of aging to have positive effects for most animals or people, right?
I think
that's it.
I think the right dose of rapamycin would have a repeatable, very statistically significant effect. And then the question is, right, if there's this paradigm where as you move sort of up to longer lived organisms, what you can still gain appears to kind of start to level out sometimes.
Yeah, I mean, that's an interesting...
So like, what are you going to are you going to get 10 years if you had just the right dose? I mean, that's not too bad. It's better than anything we've got so far, right? I think when this is understood as well as some biology is understood, when that will be is another question. You'll get a lot more than 10 years. Yeah. But I think rapamycin could buy you a few years if you knew the right dose.
Okay, so rapamycin's a 1 for laboratory animals, 1 or a 2 for humans. Okay, resveratrol, laboratory animals.
I wouldn't buy resveratrol to try to live longer, I guess is what I would say. But I don't know the latest literature, I would be honest and say that.
Okay, people. Same answer?
I guess, yeah. I would say that I'm not familiar with the latest literature in resveratrol. But
Come on, give it a number.
It's not as, I think it isn't as clear a story as rapamycin.
All right, metformin.
I think so, yeah. I think my guess is metformin, it's got a more modest possible side effect profile, so there's probably a wider range of
Are we
doses.
talking laboratory animals or people?
This is all for, people want to go get some.
Okay.
So I think that it would be hard to take the wrong dose of metformin.
Maybe we should just do people.
But I think that, I think if I had to guess, tell me if this sounds crazy because this is probably closer to your wheelhouse than mine, but that your best case effect size is probably more modest for metformin than rapamycin.
Yeah. I mean, again, my view on metformin is if you're diabetic or metabolically challenged, it's a good drug. I personally don't think there's much evidence for people who don't have metabolic dysfunction for metformin.
Okay. Is acarbose going to be in there in the same department?
You want to go to acarbose?
Well, I mean, there's a lot of data in humans for it, right? It's safe and tolerated, and
some gastrointestinal side effects, but
very much so, right? And so what made me think of that is that it probably has a more clear benefit if your BMI is a little higher or you're tending toward possible metabolic.
And again, that's
symptoms.
a diabetes drug, so
yeah. So again.
But you believe the mouse data for acarbose?
I do believe the mouse data. Okay. I mean, I think that it's kind of the Wild West in knowing. It's a bit of a leap of faith to know because we've got a lot of things that work in mice that also work in things that are much more distantly related to mice than we are. So you want to be optimistic, but we just don't have the real human data. So
yeah, I agree.
People
it's
that
hard
want to be
know.
a wet blanket, you can't really.
Right. All right. Alpha-ketoglutarate.
Some data out there that looks all right. So, the data is.
So I'm taking that these are all like 3s where you're kind of waffling.
Well, I just think that, I haven't caught up with the latest stuff on some of that, but I would say that my view of alpha-ketoglutarate is on the rise, right? I've seen more data over time about
NAD.
Less convinced.
Okay.
But it depends on which part of it you're looking at, right? But I think
less convinced is okay.
In terms of like what you could take now, I'm not sure that you could take something and expect an effect, but I don't know. Parabiosis. I think that would work. This is what I always think. I'm like, if you're like a 50-year-old guy and you get 18-year-old blood transfusions, you're going to feel like $1 million because your testosterone is going to go through the roof. But I think that it's clear that there appears to be something in the parabiosis.
I would agree with that,
but like
too.
which thing there is that's actually administered.
Yeah. Do you know this E5 story? So this is Harold Katcher's extracellular vesicles from piglets injected into rats, and apparently has help.
No,
you don't know. It's okay. It's a related to parabiosis. Just the idea that there are in exosomes.
I've
components.
Seen people talking about exosomes.
Yeah,
like I know
he calls it E5.
Yeah, so yeah, you're gonna get me in trouble with all the people from the different interventions.
Caloric restriction.
Yeah, obviously.
For people,
that's another question, because what would you have to eat, right? I like the old saying, right? It's you might not live longer, but it'll feel like it.
Right.
I mean,
right.
You'd have to eat like 1,200 calories a day or something, right?
But in laboratory animals, no question, that's the gold standard.
I mean, it is. I think it's right, and I think just like I say, like if none of these compounds we're working on turn out to be themselves translatable, we're understanding something new about how you can make something live longer that maybe we can steal from. So even if you don't want to go around eating 1,200 calories a day, if we understood why it is that these guys live longer, then there's something we can translate.
Right.
So.
Protein restriction.
I worry about that one in the sense that I think that for most older humans, I think sarcopenia is a greater risk. So I think, I think the muscle frailty that would come from
I didn't prep him at all. You're saying exactly the same thing that I say when it comes to protein restriction.
I gave one of those sort of like general audience talks, and someone asked me because they hear about this, and I'm like, so I would say protein restriction probably increases lifespan in the data we've seen, but that if you're a person and you try to restrict your protein
in mice, you mean it increases lifespan?
In the models that have been looked at, that data seems
I agree.
compelling, and it's pretty repeatable, and it's still getting looked at for good reason. But I think that what I would worry about in elderly people is that you might do more harm than good.
Yeah, and I mean, my understanding at least is that mice they certainly don't die from sarcopenia and loss of bone density, and it's probably not limiting their quality of life. Whereas in people, we know that's a pretty big concern in the elderly. So this just may be a case where even if protein restriction slowed aging in people, there might still be consequences that are detrimental that would offset the benefits. I just don't know.
I mean, none of these are the thing where what you want from the field is the thing where it like doubles mouse lifespan, and then you're like, well, we will get this.
That's what I've been saying for a long time. I mean,
right. And so I think even the best things that you could pull from what's known now are some of them are very likely to add a few years to people who apply them correctly.
Yeah.
But they should pale in comparison to what we are aspiring to as a field, I hope.
Yeah. So actually, we can stop here. We've gotten through most of the big ones, although I'll ask you a couple more. But you brought up something that I talk about a lot, and I'd be interested in your thoughts, which is, why aren't we finding things that are bigger and better? Like if you look at the mouse longevity literature, right, the longest, non-developmental intervention for lifespan is a caloric restriction experiment done 40 years ago. In terms of magnitude of effect, why aren't we finding things that are better than that? Is it because there is nothing better?
No.
So tell me why.
Tell me why.
Why aren't we finding bigger and better longevity interventions?
I mean, we're kind of stumbling around in the dark, I would say. You know what I mean? You're right.
Are we, or are we stumbling around under the lamppost only looking at what we think we already know? I don't know.
Yeah. So, I mean, I would say, right, if you think about the plasticity of lifespan across divergent species that had a common ancestor, you can see these enormous effects, right?
Yes, so absolutely. So you're talking about going across the animal kingdom and look. Yeah,
you think a mouse and a bat,
100-fold.
Back in the day, they were a mouse bat or whatever. Yeah. And something that is in their DNA, meaning we can read it out and figure it out hypothetically.
Yep.
Would tell you why one lives 30 times longer or 15 times longer than the other one. And right, so that is one of the things that people have known for a long time. Mostly they were ignored until some recent data kept reminding them of it, to tell you that aging is quite plastic, right? Yeah. And the other thing I think is the immortality of the germline, right? I mean, those cells in your body have the exact same DNA as all of your other cells, every base the same, and they're immortal. They're part of an immortal lineage of cells. And I think, it doesn't mean that looking at them will tell you the secret, but it just tells you that there's all this potential.
I guess the thing I wonder about that is how is that any different from bacterial immortality? I mean, it's, in
other words, well, let's take a look at planaria, right? They have little cells that can keep dividing and keep dividing, and they appear to be immortal. And I don't think those are unrelated, or like Hydra.
Yeah.
So in other words, I think
I would agree that there's
Mother Nature has already made it clear that there are ways for things to age much less, and even something as simple as Peter Medawar selection shadow, without adding on a lot of great work after it. But that's just something that you can immediately wrap your head around. Tells you just because we age doesn't mean we have to age, right?
So let me ask this a different way then. Do you think it's that because the field sort of, and again, we can go back 100 years to the early protein and caloric restriction experiments, because that's kind of where the field started in terms of interventions that increase lifespan, we naturally began trying to understand the mechanisms of that way to increase lifespan, and didn't pay attention to maybe some of these other mechanisms that have much larger effects that can take you from, a lifespan of 3 weeks to a lifespan of 30 years that Mother Nature has figured out? Or is it more that just the complexity of changes needed to get that magnitude of difference are so much more complex than the single gene, single intervention approaches that we've been studying so far?
Yeah, I mean, I think that the 10,000 foot view, here's what we need to do to get to the bottom of this, of a field, has essentially nothing to do with what people on the ground do in their labs, right?
Yeah, that's a good point.
So I think, but to be more charitable, I think it is true that, like, say that you want to compare the genomes of diverged species with very different lifespans, I think we only just now are starting to really have the tools to do that compellingly, and there are people working on that sort of thing. And I think they're doing great stuff, and just the tools and the data are just becoming available to make it likely that they can start to tease some stuff out. The idea of comparing really differently lived organisms to figure out what's going on has been actively looked at for a while, but I think the kinds of tools we have now with genomics and stuff are really just exponentially improving. So
I think then the question will be, how do we get beyond correlation to demonstrating that the differences at the genomic, transcriptomic, proteomic, metabolomic are causal, right? That's hard. That is going to be that has been the limitation from these comparative biology of aging approaches so far. And I think there are opportunities, but it's going to be non-trivial.
Well, right. I mean, the biases of my training will come out perhaps in my answer, because I think that in some ways I'm a geneticist at heart, right? Yeah. And so the reason that I think there's a lot of promise in comparing these species is that the answers are in their genomes, right? So how do you figure it out? You just do the genetics, So it's like, if you look at like secretion, Randy Sheckman, you look at like Yoshinori Osumi and autophagy, you just take every brick out of that wall until there's nothing left to hide under. And now you know every gene that affects this thing. That's just not a thing you can do that anyone has been able to do with the assays we want to study. But if you get answers from comparative biology that involve genes, you just do genetics.
Yeah.
I feel like, in model organisms, then you have to hope that it's conserved. But if you look at secretion and autophagy, a couple of the examples I picked up and quite a few others, it turns out that they were taken apart in a simple model like yeast in those two cases.
Yeah.
And they work exactly the same way in humans in a lot of ways.
Yeah,
so there's a lot to be gained.
I agree with you. My one concern with that approach is, and this is what I've seen in the comparative biology of aging literature so far, is what a lot of people end up doing is they look at these exceptionally long-lived animals and they find differences, and then they immediately focus in on the differences that fit into the hallmarks of aging paradigm, right? Yeah, autophagy.
I think if you're going to do a screen,
right?
Do a screen.
Yeah, exactly. So this is where I think taking an unbiased as possible approach, saying of all of these things are different.
Right.
Let's march through one by one and see which actually give us the effect.
You don't even do it one by one. You have p-values and you march through from the best on down. You know? So in other words, I think that's right. So, right. I think that there are a lot of reasons people are incentivized to circle back. If something in that list,
right, that's how you get grants. Yeah,
right. There is a consensus that they're motivated to hew to in some ways, right? But I think if what you're doing is a screen type of an approach, staying as unbiased as you can, as long as you can, is the way to move the fastest. That door may not be open to you depending on what you're up to and how much it costs to do those experiments, right? But I think that's the way.
Okay, cool. So we won't hammer through the rest of this. Do you have any that I didn't talk about that you're excited about, other than the tRNA synthetase inhibitors?
I'm a big fan of tRNA synthetase.
Okay, but don't go buy them. Actually, I don't know if you can buy them, but even if you can, don't go buy them and start taking them now.
There's one that you can buy because it will kill bacteria, but only on your skin because you will break it down immediately. So you could eat it, but unless you're allergic to it, you won't notice. And if you are,
why does it get broken down immediately?
You have a bunch of cytochrome P450s or something that just
interesting.
And so even to give it to mice, we'd have to liposomally encapsulate.
So it's a bacterial antibiotic.
It is an FDA-approved antibiotic because it in your body.
Does it increase lifespan and C. elegans?
Yes, and it's hitting their mitochondrial tRNA synthetase.
Okay, cool.
And we do it all with dead bacteria.
Yeah.
Because otherwise they'd be quite restricted.
Yeah.
So one of the things I might guess is that when we get as far as mice, that the ones that affect the mitochondrial tRNA synthetases might have less other effects.
And presumably you could find a dose, even if it's broken down by cytochrome P450, for example, that would
I don't even know if that's
stick around. I know, I'm just, but I mean, lots of drugs are, right?
The nice thing is we've got a bunch of these things that work, so we just won't use that one.
Okay, cool.
There are things, you can find things out in the world that you don't have to scour the planet to get that are tRNA synthetase inhibitors. I don't advise taking them. Because my thoughts about things like rapamycin is that it really depends on the dose. And I would say that if you talk to the people who know the most about it in the world and ask them what dose you ought to take, you won't get the same answer twice.
I guarantee it.
That's right.
Well, you might get the same answer twice, but you'll get lots of different answers.
Right. And so I think that's the trick, right? And so eventually someone will know the answer, but we don't yet. And I say,
well, I don't know if we will. This is a different topic, but I mean, no drug ever really gets dose optimized, right? It's like it gets approved for something that works, and then everybody stops trying to optimize the dose.
Well, I don't think that's true, right? Think about steroids in competitive athletes.
Even there, I mean, it's people do all sorts of.
I don't know about steroids, but in other words, imagine there's a drug that can make you live to be 200 or something, and everybody out there is taking it.
How are you going to know it's optimal, though?
Full of guys.
But how are you going to know it's optimal? It's a 200-year experiment.
Well, yeah, you won't know quickly. You will eventually know.
I don't think so.
I don't want to wait.
We'll agree to disagree. I don't think drugs ever really get optimized unless it's a really, really tight biochemical sort of optimization.
I think the nice thing about some of these interventions is that they're a really nice adjunct to the genetic approaches to take apart what's really happening. And so even if the drugs we started with, you can't just end up and go buy a pill of them eventually. We're learning about the proteasome. And what if we look into that and that's not the whole story, and we'll figure out why these 60% increase in lifespan.
Right.
I want to know what's going on.
Right.
And we have the tools to do it now.
So is anybody else in the field studying these drugs, or you kind of got this all to yourself right now?
In terms of lifespan, well, given that we've published some stuff, I expect maybe, but I don't think there's been a ton of publications about it.
If any of you would like to support really fantastic research in the field, please consider supporting Mark's research, because we need to get this stuff going faster. And this is super exciting.
Well, I'm not getting any younger. So.
No, none of us are. Well, actually, some of us are aging in reverse, I hear. All right. Thanks, Mark. This has been a lot of fun. And thanks for kicking this off at the American Aging Association
Absolutely.
meeting.
My pleasure. Thanks for having me.